Okay, good morning. We're going to start off with kind of a review about um, the cell. So, having some issues, sorry, with my screen share here. So, um, okay. So, just kind of a review. I know we went over this a while ago, but just to kind of reiterate the cell theory, which is composed of three parts. The first being that the cell is the basic unit of life. This was developed by a botanist named uh, Matthias Schleiden in 1839 or around then. The second part is that all living things are made of cells. That was developed by a man named Theodor Schwann, again, somewhere between 1838 and 1840. And for a while, that was the only part of the cell theory that scientists had. And Schwann was actually a zoologist. Like I said, Schleiden was a botanist. And the way he figured out that everything was composed of cells is he would cut up plants and he found cells in there. So he figured that all living, that the cell is the basic unit of life. So it's the smallest thing inside the plant tissues. And Schwann was able to, as a zoologist, he studied animals, and he was able to figure out that all animals have cells, and therefore all living organisms would have cells, or at least one cell or more. And it wasn't till about roughly 10 years later that Rudolf Virchow developed the third section of the cell theory, which is that all cells come from pre-existing cells. So we know it is a three-part theory. However, it wasn't always a three-part theory. So some people could argue that there are a few new parts to the cell theory, one being that all chemical reactions take place inside the cell, so which is how they function, from creating energy to dividing, going through cell division. And that some people would also argue that there's a fifth portion of the cell theory, that the cell contains hereditary information that's passed on from parent to daughter cells. So while those last two aren't official, what you would call official parts of the cell theory, one could argue that they are. So, okay, the next part, things that all cells have in common. I know that we talked about this at the very, very beginning of the year, but we'll kind of revisit this. This is a little more broad than the, I think, 5 or 6 items that I gave you. But all cells are going to be covered by a plasma membrane. Sometimes it's called the cell membrane. Plasma membrane is just the scientific version of it. And don't forget, the membrane is the part of the cell that allows things to go in and out of the cell. It's really all it does. It does separate the inside environment from the outside, but primarily its job is to let things in and out of the cell. Second thing that all cells have in common with one another is that they contain the cytoplasm, which is that empty space inside of the cell, and the obviously the fluid inside is the cytosol. And then the third thing that all cells have in common with one another is that they contain genetic information in the form of DNA. So if you are a cell, you are made up of these three, or you have all three of these things. So, plasma membrane, cytoplasm, genetic material. So, all right. If you look at the diagram that I've given you, the hand-drawn ones, this kind of differentiates between prokaryotes and eukaryotes. The cell on the left is the prokaryote, and the cell on the right would be the eukaryote. I don't know. I can't remember if we went over this or not. But where those words come from, pro means before, U means EU, means true, and karyon, derived from Greek, means nut or kernel, actually, but we perceive that as nucleus. So a prokaryote, if you put that word together, be before the nucleus, and the eukaryote would be then a true nucleus. So that's why they are named prokaryotes and eukaryotes. So if you look at the prokaryotes there, just like we said on the previous slide, they do have a plasma membrane. They are going to have DNA. The DNA is going to be in that distinct region called the nucleoid. However, it is not encompassed by a membrane, as you can tell from the diagram that I've drawn. The DNA and the cytoplasm are in direct contact with each other. There is no envelope or anything like that. The DNA is also circular in shape. That plasmid shape, I don't know if you remember that, but it's called a plasmid because it's circular. It's not going to be helical. And it's going to have proteins, but they're not going to be tightly bound, unlike the eukaryotes that will be. So we say they're loosely associated with proteins. Like the previous slide says, it's going to have a cytoplasm also. And a few other parts that prokaryotes are going to have would be protein and RNA in the form of ribosomes. So they're going to have proteins inside of the prokaryotes, and they're also going to have ribosomes or RNA, but they're not going to have official membrane-bound organelles like your mitochondrion or your Golgi body or something like that. There's going to be no cytoskeleton, but these particular cells often have cell walls. But it's important to note that they are structurally different from a eukaryotic cell wall. Going to be more basic, more primitive. And generally, they're going to be smaller in size than the eukaryotes. Oftentimes, a prokaryotic cell is also going to be a unicellular organism. So it's just going to be that one entity. You don't really have multicellular prokaryotes. However, with eukaryotes, again, you're still going to have that plasma membrane. You are going to have DNA, just like the prokaryotes, but instead of being circular or plasmid in shape, it's going to be linear or helical, and it's going to have it's going to be intertwined really closely with proteins that are called histones. So the DNA is going to be wound around these histones. Think of it kind of like a spool of thread that's wound around an actual spool. It's not just the thread, kind of like toilet paper is usually wound around that cardboard roll. So kind of think of it like that. Except there's going to be several histones wound within the DNA. Just like the diagram shows, the DNA is going to be separated from physically separated from the cytoplasm by the nuclear envelope, so it's got a membrane. just like the prokaryotes, it does have a cytoplasm. It's going to have proteins and RNA in the form of ribosomes. But unlike the prokaryotes, they will have membrane-bound organelles. Some of them are double membrane, some of them are single membrane bound, meaning some of them are just going to have one layer of a membrane, some will have two layers of a membrane, like this, much like the cell is a double membrane, because it's got that double layer of phospholipids. It will also contain a cytoskeleton, which is a complex network of filaments, so microfilaments and microtubules, which we'll talk about here in a little bit. And some eukaryotes might have cell wall, not all. And then oftentimes these are usually bigger in size. So bacteria can be both prokaryote and eukaryote. It just depends. The more primitive bacteria, the older ones, are obviously probably going to be more prokaryotic. Sorry. And the newer ones, or what you call modern day, quote unquote, like E. coli, those are going to be eukaryotic. Generally, the prokaryotic bacteria are going to be the bacteria that live in those harsh conditions, the extremely acidic or the extremely sulfur oriented, or high in sodium, the salty environments. So with the harsh ones. Prokaryotic bacteria are older. Some of them can be as old as 3.5 billion years old. Some of them are heterotrophic, some of them are autotrophic because they can photosynthesize. Eukaryotic bacteria generally are a little bit newer at 1.5 or 1.7 billion years old. So, I mean, they're still old, but not as old as the prokaryotic bacteria. You've also got a small branch of the eukaryotic bacteria called cyanobacteria, which are very well known. They're the blue-green algae. So obviously they're going to be photosynthetic. They're going to be able to make their own food from the sun. So, okay, kind of a refresher. The fluid mosaic model. I know we talked about that one back with the organelles, but with this one, it's the model of the plasma membrane. You've got phospholipids and proteins. If you remember, we've got the integral and peripheral proteins. Those are going to be the integral proteins are going to be the ones that go all the way through the cell membrane, and the peripheral proteins are only going to be in one layer of the membrane. So like that second line says, some of the proteins will run all the way through the bilayer, meaning they're integral. Some will stick out, like the glycoproteins that help serve as cell identifiers. Some only go through the one layer. There's cell recognition proteins. There's the enzymatic proteins that are located on the inside and the outside membranes only. Sometimes the enzymatic protein is only on the outside if it's meant to create something that might help the bacteria adhere to a surface. It might be on the inside if it's helping with cell metabolism or something like that. Again, a refresher, but the fluid part of the name implies that the membrane is not static. It does move. The phospholipids generally just move within one layer. They can switch from the inside layer to the outside layer. However, they don't normally do that. Proteins can generally move also, but again, they don't typically move too much. All of these are held in place by differences in charges. So they're able to stay connected side by side due to differences in charges. But worthy to note that don't forget that water can move in and out. It's the small and uncharged molecules that can diffuse in and out of the cell membrane on their own. So yeah. All right. We're going to move on into a refresher into the actual organelles that are found in plant cells. We're going to go a little more in depth into some of the organelles that you're only going to find in plant cells, though. So the first thing that's worth noting is the plant cell usually is going to contain, well, it always is going to contain the cell wall, and it's there for structure and support for your plant cell. There are only certain enzymes that can break down the components of your cell wall, which is peptidoglycan and cellulose, and so there's only certain enzymes can break it down. And actually, I have a typo there. Only certain enzymes can break down the cell wall, and I have cellulose. I don't know if it's a typo. I'm not sure what I was thinking when I typed it, but yes, the cell wall is made of cellulose. So the enzyme that breaks down the cellulose is called cellulase. I don't know if we went over this or not when we were talking about the macromolecules like the proteins, nucleic acids, lipids, and carbs. But you can usually tell that a molecule is an enzyme with the suffix ase, A-S-E. So if you see the suffix ase, it's almost always going to be an enzyme. So cellulase is going to break down cellulose. So generally, enzymes are named after what they break down. So okay. So without, if you were able to take away the cell wall, then what was left behind, it would be called a protoplast. So that word just means it's a plant cell that doesn't have a cell wall. Plant cells are obviously going to contain DNA within a nucleus. I've got a picture for you on the next section down on the doodles. It's the one on the right. It's diagramming the nucleus. So you've got DNA. I don't believe we've gone over this, but when your cell is resting, when it's not going through cell division, we refer to your DNA as chromatin. It's loose. It's not tightly bound because it doesn't need to be. It's kind of stringy. There's still going to be histones, but it's just not going to be in that characteristic X shape. That would be the chromosome shape. That would be your X shape. And it's only in that shape when the cell is going through cell division. So that's why I've drawn the DNA kind of squiggly lined, because that would technically be the chromatin. It's not in its chromosome form yet. So with that snapshot of the drawing, that particular cell would not be going through cell division at that point, since it's at rest with the chromatin. The nucleus does have a double membrane. It's one of the few organelles that does have a double membrane, and I've drawn that there. It's the nuclear envelope. The little I don't know what those are, little spaces inside the nuclear envelope. Those are the nuclear pores, so that way the RNA, the ribosomal RNA, can leave the nucleus in the form of the small and large subunit of the ribosome, because they're made inside the nucleus, and then they're taken out through the nuclear pores and assembled in the cytoplasm. Those nuclear pores also allow for messenger RNA to leave the nucleus during transcription and translation, or protein synthesis. The nucleolus is that darker spot in there, and that's where the ribosomal RNA is assembled, put together in the form of your large and small subunit for your ribosomes. And then the space inside of the nucleus is the nucleoplasm, much like the cytoplasm. Cyto meaning cell, nucleo referring to the nucleus, plasm meaning space. Okie doke. The left cell there is that I've drawn right to the left of the cell itself is um, is a picture of a eukaryotic plant cell, but it's not obviously in the characteristic square or rectangular shapes. So just so you're aware, that's my wonderful doodle. So okay, but now we're going down to the next set of doodles, which would be your mitochondria or your mitochondrion. Singular version is mitochondrion. It's important to note that with some of your plastids in your mitochondria, um, that endosymbiotic theory that I know we talked about with the organelles, that's the idea that long ago these were originally bacteria, the mitochondria, and some of your plastids like your chloroplast. But as time went on, through some sort of symbiotic event, another bacteria engulfed these smaller bacteria, but did not break them down. Rather, it just absorbed them and used them to make energy for them, meaning the mitochondria, or to make sugars for that particular cell, which would be the chloroplast. So just don't forget about that endosymbiotic theory there. Your mitochondria is the other organelle that is double membraned, besides your nucleus. My really lovely drawing there. On the left, you can kind of see that that it's got the outside membrane, but unlike your cell membrane or the nucleus, it's not a smooth inside inner membrane. It's kind of folded on itself. The inner membrane is. The outer membrane does not have a special name, but the inner membrane does, and it's called the cristae. The inside space is not a mitochondrioplasm, like you might think, but it's rather it's called the matrix. So the matrix is the fluid inside of the mitochondria. It also contains its own set of DNA, ribosomes, and RNA. So sometimes you'll hear with not evolution, but comparative anatomies, they might say, well, this organism shares similar mitochondrial DNA. That's what they mean. It's not that they share the DNA in common that are in your nucleus, but rather the mitochondria would be very similar because that DNA is similar. So technically, all of our cells contain two different types of DNA. They contain our normal DNA that you would find in the nucleus, but they also contain mitochondrial DNA. And there's mitochondrial ribosomes to help the mitochondria make things that it needs to go through cellular respiration. So, but its main job, like I said, is to just go through cellular respiration, and it's going to take in the sugars and convert them into energy, or ATP, for your cell. So yeah. Okay. So I have like 3 notebooks here that I'm trying to make sure that I hit everything on. So, okay, plastids. Plastids are very specific to plant cells. Animal cells do not have plastids. They are the last of the organelles that's going to have a double membrane. So in plant cells, there are 3 organelles that are going to be double membrane: the nucleus, the mitochondrion, and any plastid. Your plastids, there's several different kinds of plastids. And the way that you classify them is by what pigment they produce. Generally, your plastids are either going to contain chlorophyll or carotenoid pigments. Oh, I guess that's your chloroplast. I'm sorry. There are other pigments. So backtrack, sorry. So one of the plastids would be the chloroplast, and that contains chlorophyll and carotenoid. My guess is that you know that chlorophyll is going to reflect the green pigments or the green wavelengths. Carotenoid would reflect then orange, reddish orange, a little bit of yellow. So, and just in it, I've got that they're usually located on the pigments are usually located on the edge of your cell, normally, not always, but normally. So if you were looking at a cell, the chloroplast or the carotenoid pigments would be kind of on the outside. So if you see the model that I have in my room of the plant cell, if you look at it, it actually shows that all of the chloroplasts are on the outside. There's none really on the inside. They're just kind of on the outside there, on the outer rim. So, okay, if you look at the diagram, it's going to show you kind of the anatomy of the chloroplast. So it is double membraned again, and then it's got these stacks. I only drew 3, but they can have more than 3. It's the like I said, it's not the best drawing, but one stack is called a granum, and then if you have multiple granum stacks, you don't call them granum stacks. We usually call them grana stacks. And then there's a the membrane that's connecting the grana stacks is going to be called the stromathylakoid, or stoma, sorry. Nope, stroma. Just kidding. I can't read my handwriting. Stroma thylakoid. Yeah. It's written right on the computer, but I misspelled it in my notes. So yeah, that's the membrane that's going to connect your grana stacks. I did not label it, but the inner membrane of your chloroplast is going to be called a thylakoid membrane. So, so analogous to the matrix, which is inside of the mitochondria, the stroma is the semi-fluid version of the inside of a chloroplast. So this is where you're going to find, again, DNA, RNA, ribosomes, just like you did with the mitochondria. Chloroplasts have their own DNA, RNA, ribosomes, which is why scientists thought that both the mitochondria and the chloroplast used to be separate entities, because no other organelles have their own copy of DNA, RNA, and ribosomes. So that seems that's one of the reasons why they believe the endosymbiotic theory is true. So, okay. So the job of the chloroplast is to primarily go through photosynthesis. So it's going to create chlorophyll, that pigment chlorophyll, which is non-soluble in water, or it's non-polar. So It's located in the outer membranes that are made of lipids. And some of the other smaller known jobs of your chloroplast would be to temporarily store starch in the form of starch grains. And now they do that when there's no light. So they're going to store starch temporarily when there is no light available. And then they're also going to be responsible for making some fatty acids. So, okay. We'll go with this last one. So, and then we'll stop for the day because I think that's been a lot. So, okay. The other few plastids that you can have are would be a chromoplast. I've drawn those because they come in different shapes. Your chloroplast typically comes in that traditional oval shape, but a chromoplast, if you look, they can come in all sorts of shapes and sizes. So circular, triangular, blob. I guess that's what I'd call that third one is blob. So some of them, usually they're going to contain carotenoid pigments, so the orange, orangey, reddish, that's what they're going to reflect. So it's not uncommon for chromoplasts to have been chloroplasts at one point, and then they essentially morph into being a chromoplast. So you had the chlorophyll, but for whatever reason, the plant stops producing chlorophyll and now is creating keratin or the carotenoid pigments. The example that I'm giving you is the ripening of fruits. Think about bell peppers. It seems to be, I think, the easiest one, or a tomato. Everything starts green. So the tomato, if you've ever seen a tomato plant, the tomato fruit starts off as green. And then same with bell peppers. They all start off as green bell peppers. And then once the chlorophyll stops being produced, it will start producing those carotenoid pigments, and you'll get a red tomato, or you'll get a red or orange or yellow bell pepper. The same goes for many of our angiosperm trees. They normally are going to be producing chlorophyll because you're going to have a green-leafed tree. But then as autumn comes along, chlorophyll stops being produced because your chloroplasts then transition and become chromoplasts, and then start, instead of producing the chlorophyll that they have, now start producing more of the keratin. So two of the pigments that I want you to know are keratin, which I've apparently misspelled it on this one right here. So, sorry. It should be spelled with an O. And then that would be orange. Carrot, think carrots. It's the easiest way I can remember it. And then lycopene is going to produce red pigments. If you ever look really closely at a ketchup bottle, if you haven't already seen it, they're like, it's high in lycopene. Just red pigment. So nothing spectacular. And then you've got leucoplasts. So your second type of plastid that we're going to talk about is a leucoplast. These don't have any color whatsoever. So they don't have any color, so they're colorless. Or clear, I guess you could say. With these, they don't really have membrane systems like the chloroplast does, like the stromathylakoids and the granistax and things like that. They don't really have those. There are two types of leucoplasts. There's the amyloplast and the eleoplast. Eleoplast, I believe that's how you say that one. Or elioplast, maybe. It's been a while since I've had this, so I guess it'd probably be elioplast and amyloplast. So with your amyloplast, it's going to that's going to contain starch, which makes sense. Amylase, the enzyme amylase breaks down starch, so that prefix amylo or amlio is going to infer starch or sugars. And then your elioplast is going to contain or store oil. They're going to be smaller and circular. So, and then the last thing that I'm going to share with you today is that all plastids are going to arise from a proplastid. So I've drawn that's that last picture in the bottom right corner of the diagrams. You've got this little structure that's called a proplastid, and then it's going to morph into a chromoplast, a chloroplast, or a leucoplast. Usually it's going to morph into a chloroplast or a leucoplast because the chloroplast will then eventually turn into the chromoplast, but sometimes it won't. Think of your proplastid kind of like a stem cell in humans. So, but all right, we're going to stop there. That's a lot. So, hopefully this this has been informative, but we'll pick up with some more detail with the other organelles later. So have a good day.